안녕하세요. 저는 김영훈이라고 하고요. 아, 제가 오늘 발표할 아, 그 스토리는 제가 말씀드렸듯이 제가 PhD 스튜던트 때 thesis 했던 스토리를 발표하겠습니다. 제가 한국말이 프레젠테이션 하는 게 조금 서툴어서 영어로 진행하겠습니다. 아, So, hello again, everyone. My name is Colin. I'm currently a postdoc in Dr. Ryan Netz's lab at Harvard University. Yeah, I've been really enjoying my time here, and this is my current position. In our lab right now, we study how plants make medicinal molecules and how biosynthesis actually works. So, how nature is able to build molecules that are of very medicinal importance, ecological importance, and things like that. So that's kind of where my current position is. And 2 years ago, I had just defended my PhD at MIT and biological engineering. And I spent my time at the Whitehead Institute in Dr. Jinko Wang's lab. And here in the Wang lab, I really studied how plant enzymes are able to acquire new function. And I wrote up a thesis on how they're able to acquire these functions at the genomic and the structural level. So today, I'm actually going to give a talk on my PhD work on this tracing the stepwise Darwinian evolution of a plant halogenase. And this work was actually published a few months ago as the cover story on Science Advances. And it's a tremendous honor for me to be here to be able to present to the BRIC audience. So thanks for having me back. And my journey to science really wasn't all that straightforward. When I was a kid, actually, I grew up in Korea, like many of you. And there's this drawing that I had in Yuchiwan that I wanted to apparently become a hanisa when I wanted to grow up. So this is a drawing that my parents actually dug up and found. And I guess I really was passionate about molecules that are in nature and different Korean traditional medicine and fields related to that. And like many of you guys here, I grew up taking hanya, which is, I'm sure you guys all know, Korean herbal medicine used to treat various illnesses. And growing up, I often associated hanyak with this magical pouch that oftentimes tasted very bitter. But despite its distinct bitter taste, I became fascinated by this pouch containing a cocktail of plant molecules of medicinal importance. And as you may be aware, plant molecules are actually everywhere in nature. And in fact, more than approximately 1/3 of all existing pharmaceuticals out there are actually derived from plants or other natural products that are found in this world. One example is a molecule called morphine. Morphine is produced from this opium poppy plant, and it's a very well-known pain relief drug. This is listed from the World Health Organization as one of the most essential medicine that is used to treat various type of pain associated with post-surgery and different processes and medicine like that. Another example of a plant molecule that's in the clinic is a molecule called paclitaxel. And this molecule is produced by these trees called Pacific yew tree. And paclitaxel, or also known as Taxol, is a molecule that's in the clinic right now used for chemotherapy purposes. So it's a life-saving drug that is often used in treating cancer patients. And the sad reality is that oftentimes, in order to actually bring this to the clinic and be able to use it for patients, we often have to cut down 5 to 7 mature Taxus trees in order to get like 2 to 3 grams of Taxol needed for one cancer patient. So you can kind of envision, and you certainly don't need to know chemistry in order to agree that this molecule is really complex. It's really hard to build. There's many different synthetic steps that are required to actually make this molecule. So right now, it's really unsustainable to continue to source this molecule in this way. So there needs to be an alternative way to actually produce the molecule. And we can actually approach that by performing yeast metabolic engineering, where we rewire different microbial hosts and actually start to produce these molecules in heterologous systems. However, in order for us to actually do that, we have to understand the underlying biosynthesis, different enzymes that are required to actually build this molecule, to start building it in a more sustainable approach. So actually in nature, there's millions of molecules that plants are able to synthesize. But despite that, only less than 1% of these millions of molecules out there have been explored in the entirety of their biosynthesis. This drives my central research question: how do plants actually build these specialized metabolites that they've been able to evolve for millions of years to be quite clever at building these molecules? So that's the central driving question. And while I was a PhD student, I fell in love with these plants called the moonseed plants. So this is what they look like. And they actually look like grapevines. So these are moonseed vines that basically grow along the trellis, and they're native to the northeast region of the North America. So around here in Massachusetts, there's a ton, and then also in Canada as well. And I became really fascinated by these moonseed vines because these moonseed vines are able to produce a molecule called acutамine. And acutамine has been previously explored to have selective cytotoxic activity against human cultured T cells. So they've actually shown to have some potential in developing it as an anti-leukemia drug. And also at the same time, acutамine has been explored and studied to have some memory enhancing activities when tested against Wistar rat models. So it has a biomedical relevance and potential in developing this as a multifunctional drug. And yeah, in addition to the biomedical importance, one thing that I was very captivated by was actually the chemical structure. So if you look at the structure of this molecule, you can see that it's tetracyclic. So it has 4 different rings here in the chemical scaffold. And also at the same time, so that really speaks to how complex the molecule is. And also at the same time, if you notice here, there is this unusual atom, a chlorine atom, which is relatively rare in organic molecules or even natural products. And I was able to actually find that this molecule was fascinating because of this chlorine atom on acutамine. So chemically, they were very fascinating. And in fact, these halogenated or chlorinated natural products are extremely rare in plants. Despite more than maybe 5,000 characterized chlorinated natural products that are found in bacteria. So for example, streptomyces make chloramphenicol, which is an antibiotic that we use in the clinic and also in the laboratory setting. And also at the same time, there's other examples where Pseudomonas syringae, which is a pathogen on leaves, they also make these antifungal a molecule called streptomycin E, which is also chlorinated. And another example that is also used in the clinic is vancomycin, which is an antibiotic. That's a natural product found in this microbe here. that's also dichlorinated as well. So these natural products are found across bacterial kingdoms, but are extremely rare in plants. There's only a handful of cases in which we find chlorination happening in plants. So because this is such a rare case, my first question that I wanted to ask during my PhD was to figure out what enzyme is responsible for the halogenation chemistry in the biosynthesis of a ketamine molecule. So what enzyme is selectively able to install chlorine atom onto the substrate in order to make a ketamine? So to look into this, I was able to find a enzyme called dichloroketamine halogenase, or short for DAH. And I won't speak too much of it. Here is a chromatogram where we actually look at the substrate dichloroketamine, and without in the negative control, you actually see the dichloroketamine here. And once you add the cofactors, you're able to see a new peak that basically is able to convert dichloroketamine into ketamine. And this was validated via NMR and mass spec. So it turns out that this enzyme actually belongs to a family of enzymes called the iron 2 oxoglutarate dependent dioxygenases, or short for iron 2OG dependent dioxygenase. And these family of enzymes are actually the second largest oxygenase family in plants. The first biggest, largest family is the cytochrome P450s. And with being the second largest, these enzymes are in particular very interesting because it can perform these late stage, site selective CH bond functionalization. So they're able to actually target a specific carbon center at very late stages of biosynthesis, and is able to site selectively install these atoms. For example, in this case, it's a neofunctionalized halogenase that's able to site selectively install this chlorine atom onto dichloroketamine. And these iron 2OG enzymes have a very particular sequence motif. So here, for example, DH is a halogenase, and the sequence motif is very interesting and helpful in that DH, the halogenase case, there is this HXG motif where the G represents glycine and the active site. Whereas all these other dioxygenases like hydroxylases, decyterases, they all have an HXD motif, which indicates that there's an aspartate in the active site. So if you look at the active site of these iron 2OG enzymes, in a hydroxylase, there's this aspartate ion, aspartate residue that is able to coordinate the iron ion in the reaction center, which is also coordinated by two catalytic histidines along with a cofactor in 2-oxoglutarate. However, in the halogenase case, this aspartate residue is mutated to a glycine, which actually allows for an open binding pocket for a chloride atom to coordinate the iron, which allows for this rebound reaction where instead of a hydroxyl group or any other reaction occurring, it's able to install this highly reactive chloride or anion to the target carbon center. So in this initial paper, we actually found this enzyme, and we were actually able to substitute the chloride anion with a azide. So you can actually substitute that molecule that binds to the iron center with other anions, for example, an azide, and we're able to actually engineer this enzyme mechanistically to form an azidated molecule, which has downstream implications for click chemistry. So this is indicated by this new peak that actually arises when you substitute the chloride atom with an azide atom. So this paper was actually published back in 2020, and I was able to actually give a brief brick presentation back then during COVID. So you can feel free to scan this QR code to listen more about this interview. But the most fascinating part and the exciting part of this discovery was that this DAH enzyme is actually the first characterized iron 2OG halogenase of plant origin. And this was extremely exciting for us because we knew that plant molecules are extremely potent for medicinal purposes. And we also knew that halogenated molecules are potent in medicinal purposes. They're used to fine tune different activities. And up until now, we didn't know of any enzymes that are able to install site selectively a halogen onto a plant natural product. So this actually opens a whole set of different questions that we could ask to leverage and develop biocatalysts to expand chemical space and be able to install halogen atoms onto different plant molecules. So in brief summary, what this enzyme is actually able to do, these iron 2OG enzymes are able to do, is they can actually take a carbon center and abstract a hydrogen and able to install chlorine or any different like halogen atoms on there. And we've also been able to showcase that you can add azides or you can also nitrate them. So you can add nitro groups as well. So you can start to decorate your molecules at a very, very late stage part of pharmaceutical development. And this is in particular very practical and has a lot of potential because these sp3 carbons are otherwise really hard to activate. So these enzymes are able to actually allow for us to develop cool catalysts that can really target these really hard to activate carbon centers. So what if we can actually take these plant medicinal molecules and engineer plant halogenases and start to derivatize these new to nature medicine? So this is kind of the big picture of why we wanted to study the evolution of this really rare halogenase. So the question that I wanted to answer in my PhD was, how did this rare plant halogenase, the DAH enzyme, actually evolve in the moonseed plant? You see, there's millions of molecules that plants are able to make, and plant metabolism is extremely vast. But how did this event that led to the neo-functionalization of this halogenase enzyme come to be in this moonseed plant? So in order for us to actually answer this question, what we did was we took the moonseed plant and we sequenced the genome, and we were able to assemble a chromosomal level assembly of the genome. And as you may be aware, plant genomes are highly repetitive, and they're also extremely large in comparison to bacterial genomes and things like that. And it also encompasses and it captures a lot of genomic information in which we can actually mine and zoom into different parts of the location of the genome to understand more about the evolution of these biosynthetic genes. So I won't go into details of these assembly statistics. Hopefully you can just trust that the quality of the genome was sufficient enough for us to actually look into these evolutionary questions. And what we could do is we can take these high quality genome and we can perform comparative genomics and also estimate divergence calculations based off of other related, closely related species. So this is a divergence phylogenomic tree of Ranunculalis order. So these are different plants that are in the same order as the moonseed plant. So moonseed is here, Menispermum canadense, which produces this acutamine molecule that's chlorinated. And then I spoke to you about opium poppy that makes morphine, which is Papaver somnifera. And these two plants are actually in the same order and diverged about a little more than 100 million years ago. Another plant called the Chinese gold thread, or Coptis chinensis, makes a molecule called berberine, and this is one of the commonly used bioactive ingredient actually in hanyak as well. This berberine molecule has anti-inflammatory type of effect. But I speak to you about all these species in this order because in all these other families, there is no halogenation occurring. There's only halogenation occurring very specific to the Menisparmaceae family, which is the moonseed family. And if you actually look at the genomic locus of where that DAH halogenase gene is in the moonseed genome, and actually compare it with the opium poppy and the Chinese gold thread genome, and perform what's known as the synteny analysis, where you look at exactly in the genome where DAH is located, and then compare it to these other genomes of opium poppy and Chinese gold thread, and trace where this locus is located. You can actually see that DAH has no trace to these other two organisms that don't have any halogenation. So this actually goes to show that DAH is a novel gene that came to evolve in the moonseed genome. When we examine closely at the genomic locus of this DAH gene, two genes left to DAH, we found that there is a gene called FLS, and this is short for a gene called flavanol synthase. And flavanol synthases have this desaturase activity where it's able to convert a dihydrocamphorol, dihydroflavanols, and be able to desaturate this into producing flavanols, which are ubiquitously found across different angiosperms. So these are all found and extremely well conserved across different angiosperms. And we were able to actually purify this enzyme and perform in vitro assay. And when we fed dihydrocamphorol, it was able to convert it into camphorol. So it had this 2OG dependent activity. And flavanol synthase is actually traced to the genomic locus found in opium poppy and Chinese gold thread, which continues to kind of support this evidence that flavanol synthase is conserved across ranunculales plants. And at this point, we were kind of curious to see whether if flavanol synthase, or FLS, is the progenitor gene to the DAH activity. And to remind you, so flavanol synthase belongs to more of the conventional or canonical iron 2OG family of enzymes. They have this aspartate in the active site, which has this hydroxylase-like motif, whereas DAH has this glycine in the active site. So we had kind of hypothesized at this point and questioned whether if flavanol synthase could actually be leveraged to hydroxylate or act on the substrate of DAH. So we fed in dichloroquetamine, which is the native substrate of DAH, and we searched for different products that are hydroxylated or chlorinated or even desaturated. But unfortunately, we actually didn't detect anything novel. It didn't have any activity on dichloroquetamine. So at this point, the story kind of felt a little bit unclear. We knew that FLS is actually very proximal to DAH, but we don't have enough sufficient evidence to actually show that it's the progenitor gene that gave rise to the DAH gene eventually. We looked further into what is actually happening in the moonseed genome. And what we know about other closely related genomes is that in opium poppy, there's two family-specific whole genome duplication events that gave rise to duplicated genes in biosynthesis. So some biosynthetic genes that are part of morphine biosynthesis, for example, may have duplicated over the course of millions years of evolution. And we wanted to see whether if moonseed genome also had similar type of whole genome duplication events. So we did this by looking at the synonymous mutation rates, and we were able to estimate the event of whole genome duplications based off of this. And we were able to show that in the moonseed genome, there's also two specific whole genome duplications that happened millions of years ago. One of them happened to be a Menosporomaceae family-specific whole genome duplication, and then a more recent duplication event, whole genome duplication event that happened in the Menospermum genus that is very specific to these moonseed plants. So looking back at how plant genomes could actually undergo whole genome duplications, and these are relatively common in plant evolution to give rise to new biosynthetic genes, and it's an opportunity for these new genes to actually acquire new function. We were able to actually see that the chromosome that has our DH gene was also whole genome duplicated to a different set of chromosomes. So if you go back to the genomic locus that has FLS and DH, and if you perform microsynteny analysis within the same organism in the moonseed plant, we see that chromosome 3, this genomic locus, was also traced to a region in chromosome 2, where actually 3 different genes have a very similar syntenic ortholog, or in this case, I guess, paralog to a gene in chromosome 2. And when we actually looked closely, this gene was 89% identical to DH. So this is a gene that is very closely related to the DH gene and not as closely related to FLS. And we found that this gene also has a aspartate residue in the active site. So it has this hydroxylase-like motif, which is why we actually named it. And to just give a reference, FLS is 64% identical to DH. And we nicknamed this new gene on chromosome 2 DHY-like because we thought it would be a dichlorocutamine hydroxylase-like type of gene that actually potentially used to hydroxylate dichlorocutamine in the past. But we purified this gene and we tried to perform in vitro assay on the substrate of DH. It also didn't have any activity, and it was also really difficult to purify this enzyme. And another observation that we actually made earlier on, but now I'm just telling you as a story, is that there is actually a gene on chromosome 3 right next to DH. And this gene is actually 95% identical to DH. And this gene, in comparison to DHY-like gene, actually has a glycine in the active site, which allowed us to kind of speculate whether if it's a functional halogenase-like gene or whether if it's just pseudogenized gene. So we named this gene DH-like gene. And what we actually did was we looked at the sequence alignment of all these 4 genes. So I'm just showing you FLS, DH-like, DH, and DHY-like genes. And you can see that in these 2 genes, you see the glycine and the active site, whereas FLS and DHY-like, you see the aspartate. And we noticed that in this first pseudogenized gene that has the glycine in the active site, there is an internal truncation that actually caused it to lose function. So this was as a result or the cause of a pseudogenization of the evolutionary intermediate. And along with that, we noticed that in cDNA sequencing of DHY-like gene, we actually observed a premature stop codon, which led to a really difficult time trying to purify this gene and testing it as a potential hydroxylase. So we think that these 2 evolutionary intermediates are actually a part of the evolutionary pathway from FLS going to DH. So this was really exciting. And we also showed that looking at the expression pattern of these genes, we actually see very low expression pattern of these genomic intermediates, whereas FLS and DH, we actually see a relatively high expression at the log scale here. So at this point, we see at a genomic level, in the genomic lens, that we notice that the moonseed genome actually captured a blueprint of how FLS came to evolve into DAH. And this was remarkable in that we actually were able to capture these intermediate states. And at this point, we were wondering, can you structurally evolve DAH activity from flavanol synthase, FLS? If it's truly a progenitor, if we can, it's only 64% identical. If we introduce some mutations, could we actually evolve this halogenase activity? And for sake of time, I'll kind of speed through this. We collaborated with an expert in MOF and different computational biology, structural biology lab, Heather Kulik and David Kastner at MIT Chemical Engineering. And we were able to get a structural model of the halogenase, where if you zoom into the active site, you see this iron center with 2-OGU succinate as the cofactor, and 2 histidine triads here, along with a chloride atom targeting this carbon center in dichloroketamine. And what David was able to do was, using this model, we actually could look at different active site residues, and 2 actually jumped out immediately, which is threonine 231 and asparagine 262. And these 2 residues were quite interesting to us because in a bacterial halogenase structure, we actually see a similar residue that stabilizes the oxo intermediate in the mechanism, which allows for this chloride atom to actually rebound onto the target carbon really well. So this is in the WELL-05 case where serine is able to stabilize the oxo, which is kind of similar to the threonine in our structure. And then in another case, FESD, which is a lysine halogenase, there is this asparagine also that stabilizes the intermediate. So what we did was we mutated these 2 residues and we mutated them to alanine. But unfortunately, this actually didn't kill activity. And this is largely due to the fact that, one, maybe the structural model isn't sufficient enough for us to actually predict important residues, or 2, plant halogenase structures are just very different than bacterial structures. So if you look at the overall fold is actually very different, even though the active site is relatively similar. So in order for us to actually have confidence in using these models to predict mutations, what we then did was we actually looked at the active site and looked at residues that are coordinating the substrate. And we also performed molecular dynamics and molecular simulation calculation in order for us to actually estimate and calculate which residues have the most detrimental effect for solvation energy. So we were able to actually predict some of these residues that are highly important in the integrity of the structural fold. And we were able to mutate them and do alanine scanning. And we were able to find that 7 out of these 13 residues that we predicted in the GBSA hydrogen bonding analysis was able to actually kill activity. So during this experiment, we had relatively high confidence that the QMMM optimized structure of DAH was a really good structure for us to actually work with. So what we then did was we also did the same approach for FLS, and we did a structural overlay, and we started to actually introduce mutations to flavanol synthase. So as a reminder, they're 64% identical, and this is what the flavanol synthase wild type activity is for the production of ketamine. And what we first did was we just simply mutated the active site aspartate to glycine, and we actually didn't see any activity there. We also identified a iron binding residue, so 6 amino acid changes that we introduced, and that also didn't give any activity. But on top of that, we identified 8 different amino acids in this active site loop around the substrate binding pocket. And combining those 14 mutations, that also still didn't give rise to any activity. And it was only until when we added the C-terminal 26 amino acid changes in a summation of 40 amino acid mutations on FLS that we actually started to see activity, a halogenase activity. So this is 40 mutations introduced to flavanol synthase, and that's able to actually catalyze minimal halogenation activity. But you can notice that this is actually approximately 2% of the wild type halogenase activity, which just goes to show how difficult it is to actually evolve this halogenase. So we actually have a working model for how FLS came to be to evolve into DAH. So there's a more sophisticated model presented in the paper, but in short, in summary, FLS originated and was conserved in many of these angiosperms, and it underwent a tandem duplication to give rise to a second copy of FLS. And over time, this acquired a quote unquote new function in that it acquired the C-terminal extension. And at one point in the million years of evolution, we hypothesized that this new gene that was duplicated had a potential hydroxylating type of activity on D-chloroketamine-like substrates. This continued to evolve where it underwent a whole genome duplication, and we actually see a gene loss of the second copy of FLS on chromosome 2. And this continued to evolve and allow for the substrate and iron binding pocket to be neofunctionalized. And the original gene that was the second tandem duplication of FLS continued to evolve to be more DAH-like. And over time, that gave rise to the functional DAH gene that is now actually in present time able to halogenate D-chloroketamine like substrates. So this is extremely remarkable type of evolution that we can actually capture with these genomic intermediates that are actually captured in the genome. And this catalytic switch is in particular very fascinating to me. It's able to convert from acting on flavanol-like molecules into an alkaloid molecule that has a completely different structure. So I'll kind of speed through the next few results, but we wanted to extend and adapt this evolutionary model to develop biocatalysts and halogenases to further derivatize plant natural products. And that's what we tried to do. We took several number of iron 2OG enzymes that have the hydroxylase motif. There's a ton of these. They're the second largest family in plants. And what we actually did to start off was we simply mutated the aspartate active site residue to glycine. And I'll just kind of keep it relatively brief here. We were able to have success with two different cases. First case was of flavanol 3-hydroxylase. So it originally natively takes naringenin and converts it into dihydrocamphorol. But by just mutating the aspartate to glycine, you can actually be able to install chloride and an azide. And also we were able to do this with a tropane alkaloid, hyoscyamin 6-beta hydroxylase, H6H. And we were also able to install chloride and azide onto these tropane alkaloids. But one thing to note here is that the activity is still really low. Just by simply changing the active site, which is clever, we aren't able to actually evolve a fully functional halogenase. And a lesson that we learned from the moonseed genome is that this functional switch actually happens much later on in evolution. And this is captured by the intermediates where DHY-like actually still had the aspartate residue in the active site. And then it was only later on after a series of latent mutations that are happening in the background that this aspartate ion realized to change into glycine, allowing for this active site mutation or catalytic switch to happen. So just to summarize, we were able to discover DH as the first characterized iron 2OG halogenase of plant origin. And DH evolved as a result of gene duplication and whole genome duplication and neofunctionalization of this flavanol synthase, which is this highly conserved. I hope this is convincing and that latent mutations actually potentiate difficult paths to new catalytic functions. So we're able to actually get a glimpse of how plants are able to acquire new functions in their enzymatic biocatalysts. So some future directions here, obviously, we want to utilize these evolutionary principles to feed it into models like large language models to actually design iron 2OG halogenases. And also at the same time, we want to derivatize these plant natural products using these engineered iron 2OG halogenases to greatly expand chemical space in which we can use for plant molecule therapeutics. And with that, I would like to thank the Wang Group. So I had graduated in 2023, and I was able to reunite with some of them actually. So this is Dr. Jinko Wang, my PhD advisor, has been phenomenal and has been a very good advisor for me during my training. Also with other Wang Lab members that helped as part of the study. And I would also like to thank Heather Kulik and David Kastner from the Kulik Group helping me with the structural modeling aspect. And this is a photo of me with the moonseed plant at the Harvard Arboretum. So we have these natively actually growing in the Northeast. So it was really cool to actually see these plants that I'm working with in the lab in nature. So yeah, it was really cool. And I would like to thank the funding sources. And here's a QR code if you would like to connect via LinkedIn. And with that, I would also like to thank the organizers for inviting me to give this talk. And I'm happy to take any questions.